Hi everyone, thanks for joining us today. My name's Sarah Schuett and I'm a flow cytometry specialist with Miltenyi Biotec. And today I'm going to talk to you about the MACSQuant VYB Flow Cytometer and its benefits. It's going to be the perfect instrument for any labs utilizing fluorescent protein reporters, fruit dyes, or for labs that simply want to use PE and FTSE conjugates together and not worry about as much compensation issues. So to get us started, I'm going to talk about the agenda we're going to address today. We're going to dive into the optics of the MACSQuant VYB and the small footprint of the instrument. We're going to talk about the automation of the system, our technical support and hands-on training that we provide to our customers. I'm going to give you a few real-world examples of people using the VYB for their research, and then I'm going to summarize everything for you. So to get us started, the benefit of the VYB is going to be the yellow laser. You can see on the left-hand side, we have the violet, which is the 405 nanometer laser, the yellow, which is the 561 nanometer laser, and the blue, which is the 488 nanometer laser. And together, those allow us to analyze multiple fluorescent proteins at once. The VYB incorporates all of these together to come up with 10 detection channels. 8 of those are going to be fluorescent channels. It's going to give us optimal PE sensitivity and reliable APC detection on a benchtop flow cytometer. The benefit of the 561 nanometer laser is that we're optimally exciting the PE dye. You can see here we have an excitation and emission graph of the PE signal. And so we are optimally exciting the PE and collecting its emission. Another benefit here is that we spatially separate our lasers on the MACSQuant VYB, where we're exciting the GFP or FTSE off the blue laser, the PE off the yellow laser. You can see here on the top diagram, we can see the separation between those two. On the bottom diagram, that represents what most benchtop flow cytometers implement, which is FTSE and PE, both excited from the blue laser. It introduces some compensation issues that we don't want to have to worry about as a researcher. Another benefit of our optical bench on the MACSQuant VYB is that we can simultaneously detect multiple fluorescent proteins at once. Here are histograms of the CHO cells that have been transfected with CFP, EGFP, YFP, and cherry, MK2, and PI. And you can see how easily we can differentiate these signals and how bright and easily identifiable these populations are going to be. The next thing we're going to talk about for the VYB is automation. And so before we dive into the automation of the system, I really want to bring up why automation is important in flow cytometry. We are all used to having automation in our day-to-day lives now with the implementation of these home assistants that allow us to control the temperature of our homes or play music on demand. But why is automation so important in flow cytometry? And so there was a study done in 2015 that showed that 50% of preclinical research was not able to be reproduced. And that's why automation is so important. If you look at the figures here, it shows that $28 billion worth of research in the US per year is not able to be reproduced. And so automation is going to help us improve the reproducibility of our assays. It's also going to increase our efficiency and gives us hands-on and reduce the amount of hands-on time that we're implementing into these workflows. It's going to allow us to standardize our assays within the same flow cytometer, but also across multiple flow cytometers. So when we talk about automation in flow cytometry, what do we mean? And it's simple. We're talking about automating your entire workflow from start to finish. And this is the true benefit of the MACSQuant VYB. We're talking about automating from calibration all the way down to the custom express modes, which is our data analysis. And I'm going to walk through how each one of these steps of the process can be automated using the VYB, but this is where we're going to have a benefit over other flow cytometers in the market. We're going to be able to help you increase your reproducibility and reduce your hands-on time required. So to get us started, we're going to look at our simple and easy calibrations. You can see from this video, a user scanned our calibration beads, put a drop of beads into a tube. The robotic needle arm is going to come out and dilute that sample, pipette mix it up and down, and then take a sample for acquisition. the software is going to launch our automated QC program, and we're going to be able to QC the instrument for the day. Now, the only human intervention in this process is putting a drop of beads into a tube and selecting Start Calibration on the touch screen. Really, the heart of this system is this robotic arm that you saw in the previous video. And so this robotic needle arm is going to allow us to eliminate errors. The MACSQuant VYB is a syringe pump fluidic-based system. It's going to allow us to enable volumetric counts with precision. We're going to be able to grab volumes as little as 25 microliters. We're not going to have to worry about any specific tubes to run our assays in because we're not a pressure-based system. Our carryover between samples remains at an industry low of 0.01%. And this robotic needle arm is going to ensure that we're mixing our samples properly before acquisition. Because the robotic needle arm is going to mix in a pipette action just like a human. And that's going to ensure that our samples are properly mixed and we get accurate and precise measuring of our samples. Talked a little bit about the cell counting, but if you're looking to use the VYB for cell counting, all it takes is 3 easy steps. You're going to place up to 96 samples on the rack. You're going to instruct the VYB how much volume you're going to take out of each sample, and then you're going to press start. We don't have to worry about spiking any of these samples with counting beads. You can see on the right-hand side of the screen, we did do a dilution series, and the MACSQuant VYB is extremely linear over a large dynamic range. And then we also did a comparison study between different cell counters on the market, and you can see that the CVs for the MACSQuant analyzer are extremely low. We can do robust and reliable cell counts on this instrument. We also offer versatile sample processing. So you can see that first image, we have our Chill 5 and our Chill 96. The Chill 5 is going to hold 24 fax tubes, and the Chill 96 is going to hold any standard 96 well plate that's available on the market. They're called chill because both of them are going to freeze at 4 degrees. So that means you can temperature control your samples throughout your run to ensure sample viability. We also have our single tube holder in the middle there of that picture that's going to hold a fax tube or an Eppendorf tube. And then on the right-hand side, we have our universal reagent rack that can hold 5 ml glass vials and 1 ml plastic vials. And I'll show you the auto labeling feature in the next slide. The benefit of the auto labeling is that we're going to minimize human operator error. You can see from the video that the robotic needle arm came out, took a specified volume from our 5 ml tube, added it to a specific well on a plate, did pipette mixing, and then took the sample to the instrument for acquisition. So we can use this to add reagents to our samples. If we're looking to do a live dead cell marker, we can use our viability dye here. We can also use it for antibody staining. We can also even use it to dilute our sample. Everything about this process is going to be programmable, so we can program the time of incubation if we need to. We can also program the titer. And then I did mention carryover earlier, but we do have a wash station that's part of the instrument. It's going to wash the needle on the inside and the outside, and it's going to allow us to remain at 0.01% carryover per sample, which is an industry low. So I talked about standardization of QC and sample acquisition, but what about once we get into data analysis? And so on the screen, you'll see 2 different samples, and they both exhibit sample-to-sample variation. And this is something that's very common in flow cytometry. You can see the gate that we applied to the first sample on the top doesn't make sense on the bottom sample. So what about a system that, if you had a system that would recognize that this gate was in the wrong way? Okay, but what if you had a system that would take it a step further, recognizing that the gate was placed incorrectly, but then also moving that gate and setting it at an appropriate place? And this is what we can do on the MACSQuant VYB with our express modes. So express modes are going to offer a robust and standardized way to set up your instrument, to do your sample measurement, and then also to do your data analysis. And all of this is going to happen at the push of a button. So express modes are going to be automated, reliable, and simple. And basically what we do is we have a predefined experiment set up, including acquisition and analysis settings, and we're going to machine learn the data in order to apply algorithm-based gating so that it makes sense for your data set. And it's going to be automatically adjusted for each data file to achieve optimal results. There's no human intervention, there's no guesswork. All of this is going to be driven with mathematics. So an example of an express mode is FRET. So if anyone is doing FRET analysis, you're measuring the protein-to-protein interactions. This requires an unstained blank, an acceptor only, and a donor only sample, and then at least one FRET sample. And the software is going to do automatic gating. It's going to generate all of your dot plots. It's going to look at your intensity histograms. It's going to look at your median intensities, and it's going to take all of this information and compile it together and do automatic FRET calculations. And that's what you'll see here on the right-hand side of the screen. This is a screenshot from the MACSQuantify software where we ran the FRET express mode, and you can see that it breaks it down between the different samples that we ran, and then at the very bottom it actually calculated the FRET efficiency median. So no longer are we having to run our assay and then export all of our data into Excel to do further downstream analysis. The express modes can do this all for us automatically, and we're not having to introduce human error into this process at all. So let's say that you're doing something that's not FRET, and you still are really interested in an express mode. And that's where we can start and build you a custom express mode. So I have these images of the QC and the auto labeling and also a screenshot of the express mode to show you that if you implement an express mode or a custom express mode on your instrument, every step of the process now is going to be automated. We can build an express mode around your assay and your needs. We can use that to generate a report by a MACSQuantify software that's going to have all of the necessary information related to your assay. This is going to increase reproducibility, and it's going to automate your entire process. So I talked about data harmonization, but is that possible without looking at instrument harmonization? The express modes will help us to address the issue by providing automated data analysis to increase reproducibility, but is that the only consideration we need to think about? And the answer to that is no. We need to look at a way to assure intra-instrument performance as well as inter-instrument performance. And there are a lot of factors that go into that, but from our side, we have implemented what we call SmartGain technology to help this process. SmartGain technology is a universal setup that's going to ensure consistency across longitudinal and collaborative studies. We use beads with a known median fluorescence intensity to help harmonize instrument settings, which allow us to standardize flow assays across multiple instruments. This is going to work from test to test, from user to user, from instrument to instrument, and across sites globally. The SmartGain technology is going to give you consistency that you can count on. As far as support, we are here for you to support your work and your assays. With the VYB, you get MACSQuant Live support, which is going to connect you with a PhD level scientist 24/5 to help you with any questions that you may have. We also have technical and field application support to help you with assay design, custom automation, and express mode development. And then we also do offer hands-on training at our Miltenyi Biotec facilities. So I talked about how I'm going to present some people who are using the VYB in their research. Let's talk about how labs are using the VYB today. The first example is a group out of Stanford, engineering dynamic cell control with synthetic small molecule responsive RNA devices. And this group needed a way to easily and reversibly control the cell cycle. They suggested that a better understanding and control of the cycle may aid in more effective cell reprogramming. The current methods relied on small molecule inhibitors or nutrient deprivation methods, and these approaches can present a number of drawbacks. The group proposed using a ribosome-based gene-controlled devices to address the drawbacks. These genetic cell cycle controllers are able to inducibly and reversibly arrest up to 80% of a cell population in a chosen phase of the cell cycle and perform reliably over multiple experiments over a time frame of several weeks. So the way that they used the VYB in this process was to actually look at the DNA content. So in the schematic on the right-hand side of the screen, you can see their process where they are seeded in dishes, grown for 3 days, then they're collecting their cells, they're permalyzing them, and then they're staining them for DNA content. They get run on the VYB where they gate them for cells using forward and side scatter, then they look at only their single cells, and then they look at their DNA content and they analyze which phase of the cell cycle these cells are in. They also had a protocol with transient infections that you can see is included in the brackets. So if they did the transfections, they actually gated for their GFP and then looked at the cell cycle for those GFP positive cells. And so their conclusion was the first time synthetic RNA devices have been used to control the mammalian cell cycle. These RNA platform represents a general class of synthetic biology tools for modular, dynamic, and multi-output control over mammalian cells. The next group that's using the VYB is focused on developing a fast, economical, and high throughput platform to study chromatin biology. And so they are studying yeast. And so the way that they use the VYB in their study was to process 96 well plates of freshly induced yeast cultures for antibody labeling. The VYB was used to detect histones, screen antibodies, and for the mapping and binary crosstalk experiments. And you can see what their conclusion was after using the VYB for their research. It's on the screen. And then the final example I'm going to talk today is a group out of Singapore that was looking at P. pasteurus as a widely considered industrial workhorse for recombinant protein production. However, genomic integration has a relatively low transformation efficiency and some heterogeneity due to random integration. So what they ended up doing was transfecting yeast with blue fluorescent protein, or BFP, expressing plasmids, and then they cultured. And then they ran those cells on the VYB. So they analyzed these samples in 96 well plates, and they mapped out blue fluorescent and protein intensities on heat maps, and they did hierarchical clustering based on the relative frequencies of fluorescent events in each gate. So you can see their conclusion here and how well the VYB worked for what they were trying to accomplish. Here's just a slide about what our customers are saying. I really like how people refer to the VYB as something fast, rapid, and fully automated, and how it's very easy to use. So just to summarize it, if you had to listen to this presentation and come away with 4 things, these are the 4 things that I really want you to focus on. The walk-away capabilities of the VYB, how you can just load in your samples, import your experiment settings, and leave the staining, data acquisition, and the data analysis to the VYB. We can do easy fluorescent protein analysis because we have the violet, blue, and yellow lasers. We can look at fluorescent protein and reporter applications using mCherry, GFP, CFP, and many more. We have the flexible assay setup, meaning that we can easily and immediately switch between our tubes and our plates. We don't have to worry about hooking up any clunky HTSs to our system. Everything is just going to be placed on the rack right in front of the instrument. And then the multi-instrument alignment, we can harmonize data with collaborating labs to ensure the reproducibility of your assay.
Thank you, Sarah, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. Sarah, thank you so much. The first question is, can this equipment fit into a biosafety cabinet?
Awesome. Thanks, Serena. Yeah, this is a very compact instrument. So it's just over 2 feet wide, and then the depth and the height are just below 2 feet. So it fits perfectly in a biosafety cabinet. And we have customers who currently have it in a biosafety cabinet working with microbes, human samples, or animal products.
Fantastic. The second question is, can you tell me how the integrated column works for cell enrichment?
Yeah, so our MACSQuant VYB actually has an integrated MAX column on the system, and so that can be used for automatic pre-enrichment of rare cell populations prior to flow cytometry analysis. It's going to follow the same principle as our traditional MAX column. So briefly, you're going to label your target cells magnetically with our MAX microbeads, and then you're going to stain with the appropriate antibody conjugated fluorochromes. These labeled cells are going to be taken up by the MACSQuant and then passed over the column. The magnetically labeled cells are retained while the negative cells pass through the column and then go directly to the waste container. The magnetically labeled cells are then washed and eluted and then passed to the flow cytometer for analysis.
Fantastic. The third question we have, I have small sample volumes. What is the minimum sample volume needed for analysis?
Yeah, so because we have the syringe pump-driven system, we can go as low as 25 microliters. That's the minimum recommended volume for analysis.
Fantastic. My lab wants to start using a flow cytometer for analysis. Can you please tell me more about this training and science support?
Yeah, so that's one of the big values that we offer here at Miltenyi. So we have hands-on trainings at our Miltenyi locations, but then we can also do the real-time support with our MACSQuant Live support. That's going to connect you with a PhD level scientist, and that's available 24/5. And then we also have a field application team that will offer training on site in your lab and help you develop any flow panels. Then if you would like to move forward with a custom application development, we do have an additional team that's there to support any application needs that you may have.
Very good. Well, thank you again, Sarah, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Miltenyi Biotec, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years till 9/29/23, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.